recording the session as well. So in this lecture of molecular immunology, we will precisely talk about the very delicate connection, the very delicate meetup, or I must say a very interesting date between the two receptors, between the two proteins. The one receptor and one protein is the TCR, the T cell receptor present on the T cell. The other receptor and other protein appear on a dendritic cell, on an antigen presenting cell. That is called as the Major Histocompatibility Complex. Since it's made up of multiple proteins, that is why it's a complex. So the meeting between the TCR and the Major Histocompatibility Complex, we will going to talk mainly about this meeting. कैसे ये meeting होती है? And you can recall yourself, this is the place, this is the most sensitive event of our immune reaction, when the information from the Major Histocompatibility Complex, the information from the dendritic cells, the information from the antigen presenting cells is communicated, is passed on to the T cells. So much of the things depends on this connection. So if that connection is stable, is stronger, is viable, you can guess what will happen. The major the dendritic cells or the antigen presenting cells, which are carrying the bad things, which are carrying the information about the bad things, will be able to pass on that information to the T cells, or it can trigger the immune system. So that's why, but we will specially stick with only with the major histocompatibility complexes. Okay, so I wish that I will be able to add certain things to your mind, to your knowledge, with that major histocompatibility complex, and how they communicate with the how they are able to share things between the two types of cells. So when a T cell is produced within the immune system, when a T cell is produced within the immune system, the T cell is have no understanding about never met with any of the bad things, never met with any of the antigen. Such T cells are called as naive T cells. And there was a time when the T cells receives that information about the bad things from the antigen presenting cells. And after that, the T cells will start proliferating as well as differentiating. They start the process of differentiation and produce different types of cells. Those different types of cells are called as effector cells. The general term is effector cells. Effector cells are of many types. Like CD4 is an effector type of cells produced out of the T cell. CD8 is an effector cell produced out of the T cells. So there are the naive T cells will give rise to all these different types of T cells. They not only involve in the immune system, but they also trigger the beta cells to produce or to proliferate and to activate to produce a lot of antibodies. But you need to understand one fine difference between the T cells and the beta cells. The beta cells are supposed to have the antibodies. ये T cells के पास नहीं होती हैं. For example, the beta cells, they themselves, they do have their own. I'm not able to draw with my mouse pad perfectly, but they do have their own antibodies. The beta cells, they do have the chicken ban raha hai na? If I make a head over there, oh my god, a chicken run yaad aa gaya isko dekh. A beta cell is supposed to have their own antibodies. But that means they are able to capture the antigen by themselves. They are no longer dependent on the antigen presenting cells to receive information. So the beta cells can themselves receive the information. They can themselves capture and internalize and then present it as well. So ये फर्क आपने अपने पास याद रखना. But the T cells, unfortunately, they are totally dependent on antigen presenting cells to receive the information. Okay, and then after that, you know how they can affect the things and how they will going to fight. What type of affected T cells? What type of T cell is better? in a certain circumstances, depending on the nature of the infection. And the role of the major histocompatibility complex is to connect these two worlds. Remember, one world or one type of cells are receiving information from outside, antigen presenting cells. They are capturing the bad things, they destroyed it, they make small pieces and fragments which are called as antigens, and then they present it on their surface, and that is they present it with the help of the major histocompatibility complex. There are different types of major histocompatibility complexes we will discuss. They communicate with different types of T cells, so they are able to present the information on their head. Now that information which is present on the major histocompatibility complex should be communicated, should be passed on to the T cells through the T cell receptor. So the connection between the antigen presenting cell, and the T cell is the place which is the focus of this lecture. It appears like this. If you're able to see, it appears like this. This is the major histocompatibility complex. If it is not drawn here, you should be able to guess कि ये कौन से cell के surface पे होगा. You should be able to guess which type of cell we are talking about. And this is supposed to be an antigen presenting cells or an APC. Okay, because the major histocompatibility complex will be present on the surface of the antigen presenting cells. There are many different antigen presenting cells. So that is why, and different types of antigen presenting cells are required to communicate with different types of T cells. That is why the major histocompatibility complex have different types as well. They has different types. So major histocompatibility complex one side, and the other member, you can guess, if the cell is not drawn here, you can guess it is present on the surface of the T cell. The other member is the T cell. So the connection between an antigen presenting cell and the T cell, that connection is made because of the two members. You need to understand the dimensions, because the stability of this connection, the viability of this connection, the strength of this connection will define whether that information will be communicated or not. Just for instance, imagine if that connection is not made, or if that connection is not strong enough, if that connection is not stable, the antigen presenting cells will fail to pass on the information which might be very critical for the cell, fail to pass on that information. उसने पकड़ लिया, capture कर लिया bad thing, but they fail to pass on that information to the T cell. So that means the immune system will not be triggered. Or if the connection is not very strong, not very stable, the antigen presenting cells will not going to trigger the T cells in a way it should be. So the immune system or immune reaction will be less. So this is a very delicate stage of the immune system where the information should be passed on perfectly, and that is only possible if that connection is stable and that connection is viable. Certain things you need to know, that the major histocompatibility complex has different domains. Remember, the pocket of the major histocompatibility complex, this pocket, which is carrying some external fragment. This is the external fragment. This is the external fragment. You can see it has different shape. This is a cross here, and a cone here, and a circle here, and you can see a cross here, and you can see a square here. So like every antigen is the part of that bad thing. It came from the bacteria, it came from the virus. This thing came from the virus. That is why it is an antigen. So the major histocompatibility complex and the shape of the pocket, remember, the shape of the pocket will define whether the major histocompatibility complex able to capture or to bind that antigen or not. So you can see different types of shapes. There is no universal shape of the pocket. Remember, एक shape नहीं होती है. Otherwise, you can guess. अगर major histocompatibility complex की shape, pocket shape, if identical in all the cells, they will not be able to present different types of antigens. In order to present different types of antigens, it is required that their shape should be variable. The pocket shape should be variable, so that it's a variable type of thing. So in this particular case, it able to capture, or it able to present that thing, that antigen which comes from the bad thing. And T cell receptor will be able to bind themselves with the major histocompatibility complex. The reason why it is called as major histocompatibility complex is that because the MHC is made up of multiple proteins, multiple receptors. So one of the receptor is like carrying the antigen, the other receptor is supporting it or support the binding. The other receptor is supporting in the activation. So there are different types of proteins present at that juncture. That is why it is called as the complex. And that the shape is variable, and it is like polymorphic. It has like different combinations to create different types of shapes at the pocket. So it can present different types of antigens as well. Okay, so you need to understand from this chart, and it's quite simple, that MSC dependent antigen recognition. Remember, the antigen recognition is not only dependent on this thing. There are several cells which can recognize the bad thing by themselves. They are no longer dependent on MSC combinations like beta cells. They can get the information by themselves. So agar it is like an antigen recognition by T lymphocytes. So T cells, most T cells recognize peptides. Most of them, they recognize the peptides coming from the bad thing, coming from the proteins of the bacteria, coming from the proteins of the virus. Okay, so it can connect themselves with the peptide present on the MHC. Different types of this is critical to understand. Different types of protein domains are sensed differently at this connection, because agar bacteria ek qisam ka hai, that will going to produce different type of proteins, it was carrying a different type of proteins. If a bacteria is of different type, it will carrying entirely different type of proteins. So those peptides will have a different shape and will be recognized differently at this junction. Sometimes they do recognize other sort of molecules as well in addition to the peptides. Particularly, this is famously known is a place where the peptides from the bad things, from the antigen, from the bacteria, from the virus is recognized. Okay, this is important. CD4 and CD8 T cells, they preferentially recognize antigens sampled from the extracellular and cytosolic pools. I will talk specifically on this topic. Yeah, we will going to study this one in detail in this chapter. The CD4 cells are famously designed. They are famously known to be designed in such a way they can capture the peptides which are coming from outside. अब आपने यहाँ पर समझ लिया ना, ये वाली तो धन्य हो जाएंगे हम. We will be very happy अगर हमें समझ आ जाए. Okay, CD4 cells, they are designed to talk with the MHC, which is carrying extracellular proteins, the proteins which are captured by the antigen presenting cells from outside. अब guess करें, the antigen presenting cells will capture the proteins from outside, means the proteins from bacteria which are coming from outside. Full stop. The CD8 cells on the other side, the CD8 cells are designed to talk with the MSC, with such MSC which are carrying the cytosolic proteins, the proteins which are produced within the cells, the bad proteins produced within the cells. So you might be wondering, bad protein produced within the cells, what that mean? Why the cell is producing the bad protein, and why it need to present on the surface, and why our cells should feel bad about the proteins which are produced in our own cytoplasm. So think about the virus, the coronavirus, and the other similar sort of viruses. The viruses, when they penetrate into the cell, they integrate themselves into the human genome, and they push our own translation machinery to start producing the proteins related to the virus. Now, the human cell will start accumulating the proteins of the virus. ये प्रोटीन एक्स्ट्रासेलुलर है कि इंट्रासेलुलर है? Such proteins are intracellular, cytosolic, produced in the cytoplasm. So those proteins will be presented in a different type of MSC, and that protein will be recognized by a different type of T cell. कौन सा वाला T cell recognize करेगा इसको? CD8. And this thing will happen only during the viral infection. But that means if the virus infect a cell, the cell will going to start producing the viral proteins. It will be presented by a different type of MSC, recognized only by the CD8 cells. The CD8 cells are activated when the CD8 cells will be activated when there is a virus infection. The last three topics, lectures, sorry, the papers which we have talked earlier, all these papers, particularly in one paper where they want to prove, I don't remember the name of the paper, you can recall, where they want to prove that the B2 type of vaccine is able to trigger the immune system. They specifically mention the increased level of CD8 cells. But that means they are actually convincing the rest of the world that after giving the B2 stuff, the CD8 cells got activated. Means it started feeling it as a virus, considering it as a virus, and that's like a success of their approach as well. Presented it as a success of their approach. Okay, are you with me? I wish ke abhi aap log zyada thake hue nahi hoenge. You should not be very much angry. Let me see the time. Please don't get weird with molecular immunology. And in the next slide, we will be talking about how different type of cells pick up the different antigens. We have already discussed these things. I'm just telling you some additional advanced information related to this thing. The dendritic cells, you know where the dendritic cells are located. The dendritic cells, when they capture the bad thing, they're able to internalize it, endocytosis karke isko kha lete hain. And then after that, they destroy and break down this external thing. For example, this is the bacteria. They destroy the components of bacteria and then present it on their surface. Now, if you zoom in that portion, please apne aankhon se zoom in kare. If you're able to zoom in that part, the combination, this is the dendritic cells. So this green stuff is the MSC. This green stuff is the MSC, Major Histocompatibility Complex. And if you can see a brown colored stuff present between the folds of MSC, this thing is the fragment, is the protein fragment, or I must say the peptide from where? From bacteria. Is bacteria ka ek tukda, chota sa, which is like an antigen, is present here. So this is an MSC complex. जिसने अपने सर के ऊपर उठाया हुआ है, holding up a fragment, a peptide of the antigen. And this is now presentable to the next cell. किसको present करेंगे? The dendritic cell specifically present information to the naive T cells. They are designed to talk with the naive T cells on. So the naive T cells will like hovering around here and there, nothing bad, no, everything is fine, everything is fine, everything is fine. And suddenly it touch the dendritic cell, which carries the MSC containing an antigen from a non-self thing. So that naive cell, that meeting might be taking place in the blood, in the skin, somewhere in the lymphatic system or in the lymph nodes, particularly. Yahan par zyada time hota hai, they spend a lot of time with each other. So that connection will communicate that information about that antigen to the naive T cells. But that is not enough. You can see some additional supporting connections are required. It is very important. Very important. The protein receptor present on the antigen presenting cells or the dendritic cell is called B7 receptor, is required to communicate or required to connect itself with the CD28 present on the T cells. So आपने ये confuse नहीं करना. CD28 is supposed to be present on the T cell. If you remember the concept of CD cells, CD, cluster of differentiation, we mark our T cells with different names depending on the nature of presence and absence of certain combination of receptors. So CD28 is a kind of the number given to a one particular type of receptor present on the T cell. So CD28. So it is always present on the T cell. B7 is present on the on the dendritic cell. So in addition to the MSC TCR, this is the TCR, red one, this green one is the MSC. In addition to this combination, the connection between the B7 and CD28 is required in order to push the naive T cells to proceed further with the proliferation and differentiation and to produce the effector cells. When I said effector cells, that means all sorts of T cells, CD8, CD4, helper T cells, memory cells, blah, 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 whatever. But the naive T cells will only be triggered if that thing is like, that connection is established. So the people who are not able to show a stronger response against a particular bimari, against a particular disease or a particular antigen, the reason might be anywhere. I mean, there are lots of reasons you can guess. At the end of the class, you might be wondering why we are not getting sick on every day. So one of the reasons is that the antigen presenting, this connection is not stable. If this connection is not stable, the dendritic cells might not be able to pass on, might not be able to kick the naive T cells to start proliferating and produce lots of response like effector cells. Other cells which can communicate that information, they are macrophages. The macrophages, they can capture, they can internalize, they can broken down the fragments as well. And after that, they can present the fragments in the same way to the effector T cells. The difference between the macrophage and dendritic cells is that the dendritic cells are supposed to share that information with the naive T cells. And the macrophages, they are supposed to share that information with the effector T cells. If the effector T cells are there, that means the trouble is there. Your body has already understood about the infection. They came to know about the infection. So this information coming from the macrophage means the information is quite big. I mean, the macrophage wants to get activated. The macrophage wants that effector T cells should produce more response by activating the macrophages and by activating the beta cells. Look at that. What is meant by the activation of macrophages? ये हमने पढ़ा हुआ है classical activation. I wish you remember that thing. Once the macrophage shared that information with the effector T cells will notice, oh my God, this is the same antigen I just learned from the naive. When I was naive, I came to know this is the bad thing, and you are telling me the same information. You are giving me the same information. So that means the trouble is real. The trouble is huge. So how about, so then the effector T cell will start producing the chemicals as well, cytokines. Those cytokines will activate the macrophages. Activation of macrophages means it will start producing more lysosomes. It will start capturing more bacteria. It will start producing more MSCs. So that is why the function of the macrophage will be enhanced. जैसे अगर पहले, for example, it is capturing earlier, it was capturing 1 bacteria per cell. Now it will start capturing like 5 bacteria per cell, just pushing and triggering the macrophages. In another situation, yet in case of beta cells, the beta cells, they do carry the information on the surface. They can read with the help of the antibodies they are carrying. So they can capture the bad thing. They capture the bad things, especially the things which are flowing in the blood, things which are like present in the blood, humoral immunity. So they can capture, they can recognize, oh, something weird is going on, something weird is present in the bloodstream. They can capture, internalize, and then they can present it to the T cells as well. But they're not going to present it to the naive T cells. The naive T cells will only receive the information from the dendritic cells. So that is how they're able to activate, they share the information with the effector T cells. Effector T cells is a general term, but I can tell you, please remember, this effector T cell is called as helper T cell. इसको बोलते हैं helper T cell, or more accurately CD4, if you remember, helper T cells. Paper में भी था. The helper T cells, the CD4 cells, are the ones which can talk with the beta cells, activate the beta cells, and once the beta cells got activated, it will produce the plasma cells. The plasma cells are designed to produce antibodies. So think about a very bad situation of a disease. The naive T cells receive it from the dendritic cells. The dendritic cells are like moving around like rangers in different organs of our body. They get they sense it information. They produce effector cells like helper T cells as well. One of the cells get the same information from the macrophages as well. They kick off the macrophage to start killing the same thing with a better efficiency, And at the same time, if the affected T cells came to know about the same trouble from the beta cell as well, that means that infection is present everywhere. It is present in the blood as well. So they need a very massive response. So the massive response is only possible when the beta cells are triggered to produce antibodies. So that is the kind of state our body is experiencing. So these are the different type of cell types, and they are carrying different types of MSC. Generally, you need to know about the three types of MSCs: MSC1, MSC2, and MSC3. They are communicating between different types of cells. But the most famous of them is the class 2, which works most of the time, the class 2 MSC. And dendritic cells are communicating through it, macrophages, beta lymphocytes, and other cells. They are communicating with the help of those things. And they need some additional support as well, that is called as the co-stimulators. I just shared one of the co-stimulator with you. Can you remember this one? This is the stimulator, co-stimulator. The binding is done between MSC and TCR, full stop, but it is supported by the V7 and CD28, and this is also called as the co-stimulator. The co-stimulation push and trigger, the co-stimulation push and trigger the cells to get activated. And you can guess if anything bad happens there, and anything bad happens with the molecules which are communicating, anything bad happens with the cytokines over there, that the cells will not be able to be pushed perfectly. They will not be able to get the things right. And that is why the immune system will not be able to fix the disease as well. Hope this will understand. You understand all these things. I'm just stopping myself here just for some question answers from your side. If you have some questions related to the things we have studied, I wish it is helping you to enhance and boost your knowledge related to molecular immunology. So I'm open for your questions.
सर आपने शेप ऑफ पॉकेट्स ऑफ एमएचसी के बारे में कुछ बताया था.
हाँ वो हम पढ़ेंगे. we will see individual class of MSC. ये जो हमारा चैप्टर है इसमें हम पढ़ेंगे. we will see MSC1, MSC2 की pocket shapes में क्या फर्क होता है and how they will be able to capture different types of antigens. ठीक है, ये टॉपिक है, don't worry.
सर जब APCs इंफॉर्मेशन पास ऑन कर देते हैं टी सेल्स को, तो एंटीजन जो है उनकी सरफेस पे अटैच्ड रहता है.
Yes.
तो वो फिर एंटीबॉडीज जो स्पेसिफिक प्रोड्यूस हुई हैं वो उन APCs को टारगेट करती
उन APCs को टारगेट करती हैं.
क्योंकि उनके ऊपर वो एंटीजन.
Okay, it's a good question. कौन है भला ये? Who is that?
सर
किसने पूछा मुझे सवाल? Neha, you have a wonderful imagination power, I must say. Congratulations. आप गलत फील्ड में आ गई हैं. आपको शायर होना चाहिए, लिटरेचर में जाना चाहिए. सॉरी. मैं एक्सप्लेन करता हूँ बाकी लोगों को कि नेहा ने क्या क्वेश्चन किया. नेहा ने क्वेश्चन ये किया है कि एपीसी ने प्रेजेंट किया एंटीजन को सरफेस के ऊपर. दैट इंफॉर्मेशन इज कम्युनिकेटेड टू द टी सेल. द टी सेल गॉट एक्टिवेटेड. फुल स्टॉप. गल समझ आ गई. दैट्स ऑल. नाउ what will happen with the APC जिसने उठा के रखा हुआ सर के ऊपर एंटीजन? I mean about the bad thing. So when the T cells got activated, they will produce effector cells. They will push the beta cells to produce antibodies. Now those antibodies are against the antigens. The antigens are present where? The antigens are present in the bad thing, I mean in the bacteria. So those antibodies will going to kill the bacteria. But those antibodies are also present. Those antigens are also present on the surface of APC. क्योंकि APC ने पकड़ा हुआ है उसको. So those antibodies will not only kill the bacteria, but they will also kill the APCs, which are like जिन्होंने इसको उठाया हुआ है. The answer might be yes, but I don't. What I can tell you is that our immune cells, they have address, they have different tags. Our immune cells, they are carrying different tags. ओही मैं अपना हूँ, मैं आपका अपना फौजी हूँ. Okay, so we will see those tags as well. There are tags on our cells which allows or help our immune system to recognize between the dost and the dushman, to recognize between the friends and the foe. अल्लाह करे ये हमारे पास भी हो. वैसे मैंने एक लिखा था उर्दू में कि we should be able to recognize like in couple of एक week में हमारा immune system पता लगा लेता ना who is our friend and who is not. So the cancer cells, they use the same trick. कैंसर सेल इसी ट्रिक को इस्तेमाल करके चीट करते हैं आवर इम्यून सिस्टम, बिकॉज आवर इम्यून सिस्टम शुड किल द बैड सेल, बट द बैड सेल इज कैरिंग द टैग्स. वो जब उसको देखते हैं यार ये बुरी चीजें प्रोड्यूस कर रहा है, बट द टैग इज ये तो मेरा अपना सेल है, so they do not attack on those cells. But even I don't know the exact answer, but even then if they start killing the APC, that's exactly fine, because those APCs have done their job. They actually communicated the information and they will kept on communicating the information. एक T cells को trigger किया, the other T cells को trigger किया, फिर other T cells को trigger किया. That's possible. ठीक है, क्या good question. बड़ा सोचते हैं आप लोग यार, छा गए. And anybody else?
हम सर
क्या आपने कभी मूली वाली कॉफी पी है? जी
सर मैंने पूछा था कि डायग्राम में मैक्रोफेज की सरफेस पे 3 ग्रीन स्ट्रक्चर्स हैं जबकि बाकी सेल्स में सिर्फ एक एक है, तो क्या वो तीनों एमएचसीज हैं?
जी बिल्कुल बिल्कुल. और मैं अभी मुझे बड़ा मजा आता जब आप लोग सवाल पूछते हैं जिसका ताल्लुक अगले लेक्चर से होता है. I mean it's when you people will be like me or like you people will try to share your knowledge with other people. ये एक पैमाना होता है, एक टेस्ट होता है कि when your students and the people you are talking with, they are asking things which are related to the next level, so they are excited to know कि आगे क्या होना है. So I will tell you, अभी आपने एक बड़ी मजे की बात नहीं देखी, वो उससे आपका कॉन्सेप्ट क्रिस्टल क्लियर हो जाएगा अबाउट एमएससी. एमएससी बनते कैसे हैं? अभी तक आप ये समझते हैं, until now मेरे मूलियां खाने के दौरान आप ये समझते हैं कि the MSC receptors are created on the surface, लेकिन ऐसा होता नहीं है. But I can tell you the MSCs are created within the cells and then after they are exported to the surface. वो पढ़े उससे फिर क्लियर होगा कि why one cell is like carrying multiple MSCs, और एक सेल के ऊपर मल्टीपल एमएससी होंगे, वो अलग अलग होंगे, एक तरह के होंगे, दे आर प्रेजेंटिंग वन एंटीजेंस और मल्टीपल एंटीजेंस. देखो एक बैक्टीरिया को तोड़ा है आपने, उसकी किसी एक प्रोटीन को तोड़ा है, जस्ट इमेजिन देयर माइट बी 4 फ्रैगमेंट्स व्हिच आर कमिंग आउट ऑफ दैट वन बैक्टीरिया, है ना?
यस सर.
Those 4 fragments will be presented separately on the surface. Those 4 fragments are different. That is why they are able to trigger the naive cells differently. Are you with me?
यस सर.
बड़ा जबरदस्त कॉन्सेप्ट है ये, बहुत जबरदस्त कॉन्सेप्ट जो मैं बताने जा रहा हूँ. The 4 अलग टाइम नहीं है मेरे पास. The 4 different fragments of the same bacteria. एक ही बैक्टीरिया के 4 fragments बन गए. The 4 fragments are presented by 4 MSCs. The 4 MSCs will trigger the 4 different pathways of immune system. वो 4 अलग अलग किस्म के नाइव सेल्स को ट्रिगर करेंगे, 4 अलग अलग किस्म के इफेक्टर सेल्स बनेंगे. They will trigger the 4 different types of beta cells and they will going to produce 4 different types of antibodies. मेरी क्लास अगर मेरे साथ है ना तो हम छिक्का मारने वाले हैं. We're about to make a 6. अगर मैं आपसे पूछूं वो जो 4 एंटीबॉडीज बनी है, the 4 antibodies created at the end of the reaction, those 4 antibodies are targeting what? चारों का टारगेट एक है?
यस सर.
Because all of them are targeting against one bacteria. अगला सवाल, क्या ये चारों एंटीबॉडीज सेम है? No, because they are generated from the different fragments of the antigens. So in totality, the 4 antigens trigger the 4 antibodies. The 4 antibodies are different from each other, but they are targeting the same bacteria. They're targeting the different portions of the same bacteria. ठीक है?
यस सर.
That mixture, अब 6 की लाइन है. That mixture of antibodies which are different from each other, but they are targeting the same antigen. They are targeting the same pathogen. They are targeting the same antigen. Such mixture of antibodies is called as polyclonal antibodies.
राइट सर.
They are called as polyclonal. अगर आपको polyclonal antibody समझ आ गया ना, ये वाला मेरा molecular immunology पढ़ाने का objective जो है ना वो पूरा हो जाएगा. I will be very happy if you able to explain the same thing in a perfect way.